At the Ludwig-Maximilians-University in Munich, we work daily to improve our methods for identifying contributors in forensic genetic evidence. In the field of forensic genetics, there is a need to resolve cases in which cells from multiple contributors are mixed together. We use the conventional methods for differential lysis for sexual abuse samples and try to deconvolute mixed profiles using software algorithms, but this is not sufficient for resolving all cases. When we came across DEPArray technology, we thought it might enable a different strategy for resolving these cases and open new ways to handle mixed forensic samples. Sometimes both the victim and the perpetrator each contribute a different kind of cell. This is typical of sexual abuse crimes, but it's not limited to that. In these cases, isolating poor pools of cells of the same type can lead to the generation of clean profiles for each individual contributor.
Initially, there was not much information on how antigens on the surface of cells would keep their epitopes intact in forensic samples. Thus, we obtained and tested some real samples that were 10 and 27 years old from the police archives. The results were very encouraging indeed. Antibody staining worked well and allowed for precise identification of blood and epithelial cells in all samples. The STR profiles were almost complete in the 10-year-old samples, while in the 27-year-old samples many alleles had been lost due to DNA degradation. But the good news is that the few alleles still present were all of 100% concordant. Thus, even from a sample such as that, it might be possible to gather useful information like a non-exclusion.
In our first paper, we demonstrated how the isolation of poor pools of cells from a buccal swab, taken from a patient who had undergone a bone marrow transplant, led to the generation of clean STR profiles of both epithelial and blood cells. In the second paper, we worked on a real crime scene sample and we were able to couple two important pieces of information together: the type of cells and their genetic profile. We were thus able to clearly conclude that the mixed stain contained blood that belonged to the victim. In other cases, things can be more complicated, for example when the same kind of cell are left by both contributors. This is where single-cell analysis can be very helpful. We were also a bit skeptical about being able to obtain a STR profile from single cells in the beginning, but the results speak for themselves.
Single cells were never really used in forensic science before. Some people tried with laser capture microdissection, but this didn't solve any cases. It is very difficult to isolate just one cell and avoid contamination by DNA from adjacent cells. Using the DEPArray technology, it is possible and relatively easy to perform. Cells are stained with antibodies loaded on the chip and scanned by the microscope. Once cells are selected, everything is done automatically by the electrophoretic chip and the cells are delivered individually in each tube. The operator can see each single cell image and decide if it's really the cell he or she wants to collect. We can then lyse the cells and start the PCR directly in the same tube to get the STR profile.
We first tried single cells isolated from fresh blood and got an average profile completeness of 85% with very high concordance. We then tested it on a real casework sample, a knife blade that was 3 months old. The blade was stained with mixed blood and it was not possible to obtain a single profile. We processed the swab through DEPArray and obtained a series of 15 single blood cells, which we profiled one by one. Profile completeness was a bit lower than it was on fresh cells, but impressively, the dried cells preserved DNA integrity pretty well, and we were able to obtain concordant partial profiles for most of the cells. All single-cell profiles were used to reconstruct a clear profile of two contributors. We think that this can have a big impact in solving complicated cases, including gang rapes, mixed blood samples and mixed saliva samples.